This young man seems to see the future. Throughout his life, this will be the essence of Jim Watson's genius. At 23, when his bosses tell him not to work on DNA, he ignores them. He discovers the double helix and wins the Nobel Prize. When he writes a book about his life, his friends say it's too candid, but he ignores them and publishes it anyway. It's a bestseller, and he becomes a millionaire. When he begins his biggest venture, the scientific establishment tell him it's not practical, but he persuades the U.S. Congress to give him $3 billion, and now the human genome has been mapped. But today, he has a vision of the future that many find disturbing, and some people wonder if this time his judgment has finally gone off the rails.
I find myself, realizing that most people don't want to hear important truths.
DNA is the molecule that controls life. It's in every cell of every living thing. 50 years ago, almost no one knew of its existence. But today, the human race is unlocking its mysteries. And when you understand something, you can start to control it. Choosing the characteristics of future generations, screening out the genetically undesirable, designing our own DNA. The possibilities of this technology seem limitless, but perhaps there are moral limits on what should and shouldn't be done. Who should make the decisions? Where do you draw the line? We asked Jim Watson for a guided tour of the future, a look into Pandora's box with the person who opened it. We begin here, at the world's leading producer of genetically modified foods. Their desire to enhance nature makes Monsanto one of the most controversial companies in the world.
The purpose of a growth chamber is to provide an idealized environment for the plant.
For this reason, many scientists would be apprehensive about coming here. But not Jim Watson. He loves GM food.
One of the adult Colorado potato beetles is on display on that white.
Monsanto does is add genes to their plants, changing their genetic makeup to suit the farmer's needs.
We have here 2 cotton plants. This is what a cotton plant would look like in the old days. Sick and diseased, and there's nothing you want to see here. And here's one which has been genetically engineered to produce this protein which absorbs the insect guts and kills it quite fast, within a day or so. This is a perfect plant. And if you wanted, an insect-dominated world, this is it. But this is a, rational man's dominated world. And to me, this is the future.
What Watson knows is that what has been done in cotton and corn could one day be done in animals and people. But even Monsanto draws a line here.
Okay. So genetic manipulation will be more and more accepted. A lot of people don't want the world to be changed. But we shouldn't be too upset. We just have to go on with our business. We want to make the world better. There are people who say, well, we're playing God. And, I have a straightforward answer. If we don't play God, who will?
The next generation of DNA scientist, Mario Capecci.
Jim was a very important influence on my life. Literally, we live in the future.
The process begins with this machine. The bottles at the bottom are full of the ingredients of the genetic code, because this machine can make DNA.
This is an automatic DNA synthesizer, and what you can see here are the nucleotides A, G, C, and T. And so what this machine can do, we can program it to put in, as we're synthesizing the DNA, we can specifically put in A at this position, then a C at the next position, and a T and so on, and then thereby resynthesize a part of a gene. And so in essence, we have the capability of changing the genome in any way we see fit.
He then has another machine that incorporates the new piece of DNA into the mouse cell.
We put an enormous voltage jump on this, and in the process what happens is that the little holes are opened up in the cells, and the DNA slips into the cells and then goes into the nucleus and then participates in making the DNA of the mouse.
In this way, he can change the mouse embryo when it's just a small cluster of cells, and then observes how the new DNA affects the way the animal forms. This mouse has had a gene switched off so it cannot move its face muscles. It cannot blink. Now that might sound cruel, but Mario Capecci does this because there are children who are born without the ability to move their face muscles, and his research is designed to look for a cure.
99.9% of all the genes in a mouse and a human are going to be the same. So whatever actually we learn in a mouse is going to be directly applicable to humans.
He's turned his attention to chromosomes. Our DNA is wrapped up into 23 different sets, which carry our genetic traits from one generation to the next. Capecci doesn't want to tamper with any of these. He's thinking about making an extra artificial chromosome. It would carry a bundle of genetic improvements, which he could switch on and off, just like he does in mice. The artificial chromosome could even be upgraded like computer software before being passed on to the next generation. Any alteration comes with a kind of money-back guarantee.
What's wrong with it? You would say, well, give them an unfair advantage. But some people now are born with, much better abilities than others, and we don't say they shouldn't be born.
And how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine, AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly.